Hello students, I am Dr. Ashwath Narayan. In this session, we're going to discuss about plant transformation techniques. So before going to that, you need to know about what is transformation, plant transformation. The plant transformation is a technique where we'll introduce a desired gene into a host plant to get a good yield, a disease resistant, fungal resistant traits, so that we effectively produce the yield without the interference of the bacterial infection, fungal infection, and other type of infestation. So this technique is very essential in plant breeding techniques to get elite traits of the plants, or elite traits will be get because of the plant transformation techniques. If you go one by one, if you see, this is the different techniques we're going to discuss in this slide, in this discussion. We're going to thoroughly discuss about these kind of methods here in plant transformation. So plant transformation methods are directly, broadly classified into direct methods and indirect methods. What is direct method? What is indirect method? Means, directly we will insert some gene of interest. Directly we'll make it, physically we'll insert that, or chemically we can insert that directly into the target host cell. That is called as direct method. So indirect method means we'll use some type of viruses. It may be, in some type of viruses or bacteria will be infected. They have some machineries, so directly they can integrates into plant genome effectively as compared to direct methods here. So we'll use both direct and indirect method. Generally we'll use this method because it is so much effective. Why? Because Agrobacterium tumefaciens will have a TI plasmid. This TI plasmid successfully integrates into a plant genome. So it is called as a live engineer, plant engineer. It is called as. So we'll go one by one. First one is called as physical methods. Physical methods were, so first one is called as this one. The first method is called as pollen mediated transformation. What is pollen mediated transformation means? So we'll select a desired plant. We'll select a desired plant here, and then we'll collect a compatible pollen grains, and then we'll modify a genetic material of that respective pollen grain. This pollen grain contains exine, intine, the internal nucleus. It has the vegetative part and generative part will be there. Vegetative will have a vegetative nucleus, generative part will have generative nucleus, and we need to target generative nucleus in such a way that desired trait will be inserted into the generative nucleus. So that this is genetically modified pollen grain will be sprinkles onto the hand emasculated stigma of the desired host plant. Whenever you do this kind of hand emasculation, hand pollination with the with the genetically modified pollen grain, so we can get the desired trait of the plant with this method. How effective this is? This is, the percentage of effectiveness is 40%. It is effective in getting the genetically modified plant. Next method is called as pollen tube method. What is pollen tube method? Pardon me. What is pollen tube method? Pollen tube method is a method we can you can observe that uh, it is also same as that of the pollen mediated transfer, where you can observe that instead of direct pollination, instead of take a hybrid pollen grain which is genetically modified and put it on a stigma of a flower, desired flower. So we'll directly go with a, we'll insert a drop of pipette. We'll cut the style. If you take the reproductive part of female and gynecium, so this is called as stigma, this is called as style, and this is called as ovary, right? So in that, whenever when pollination happens, we'll cut the stigma up to some part of the style. Whenever you cut that, pollen tube is projected inside, and then you can observe that we'll make a micro pipette here, and then the pipette contains the a gene, a exogenous gene, genetic material directly inserted, trips onto dipped onto the pollen tube. Whenever that will be dipped onto the trickles onto the pollen tube, this drop will take up this genetic material, and then directly enters into the gynecium. Whenever that happens, definitely there may be a chance of transformation, so that these cells will modify in such a way that, assume that this solution, the pipette contains solution contains genetically modified Agrobacterium ti plasmid, and this ta plasmid contains desired genes. Whenever desired genes are there, so definitely that will directly integrate into the cell, and then we'll get definitely a transformed cell further. So this is the second way, physical method of transformation. The third one, you already know that this is called as micro injection. This is successfully happening here in case of in vitro fertilization, embryo transfer of human beings who are all suffering from sterility, infertility. They are going with this micro injection method. What they will do, they will collect the protoplast here. This is protoplast. This protoplast means plant cell without cell wall. So protoplast will be collected, and then they will hold up with the suction pressure. Some suction pressure is applied, so that it will attach, it will absorb on the surface of that, so with the suction pressure. And then we'll use some micron, 0.5 micron of or 1 micrometer of this micro injection that contains the desired gene, and we'll insert that into the cytoplasm. So directly we'll percolate into the cytoplasm. Whether we can directly inject that into the nuclear DNA, or if you want any other targets like mitochondria, other things, we need to go for microscopic observation and then target the cytoplasmic organelles here. Generally, we'll go with nucleus and then insert the gene of interest, but in some condition, we'll go and target the mitochondrion or chloroplast, that is with respect to some cytoplasmic inheritance. The third method is called macro injection. Macro injection is also like micro injection, where the tip needle of that will be bigger in size. So where whenever the suction pressure is hold up the cell, protoplast cell, what happens in this DNA, the solution that contains in the macro injection needle will be inserted into the nucleus or in the cell organelles, that is respect to bigger cells, bigger plant cells here. The third method is called as pressure mediated gene transfer. What happens, some instrument is there that will generate a high pressure. Whenever pressure is exerted, because of the pressure, it will create a tiny pore on the surface of the protoplast. This protoplast, because of the pressure, it creates a tiny hole, and then we'll dip into a solution or a polyethylene glycol solution that contains a chimeric DNA or exogenic nucleic acid will be there. A chimeric DNA is inserted that should be targeted on the nucleus or cytoplasm, that will percolate through pores and then enter into cytoplasm as well as the nucleus here. You can call, you can ask me, sir, whenever exogenous nucleic acid enters, whatever the cell, in cytoplasm there is nucleases, right? Nucleases, DNases, RNases are there that will degrade the naked DNA, right? So instead of, what happens, we will make up with some associated proteins here. Whenever proteins are associated, or even we can make a liposome here. Liposome on the surrounding, we can make a surrounding of liposome that will enters into the nucleus, and then there is no chance of degradation. So that's in this way we can achieve the gene transfer. This is also one of the uh, rarely used mechanism. Next one is called as vertexing. Vertexing is not commonly used, but whenever some cells not able to take up the Agrobacterium tumefaciens type of gene transfer or gene transfection, we will use some other methods here, alternative methods like vertexing. What we will do means, solution like, calcium chloride or polyethylene glycol is mixed with a chimeric DNA, right? And then one more thing we'll insert is silica bead or other type of beads are used. So these beads will, which on the surface of that, it absorb a chimeric DNA. Whenever when in a machine called as vertexing machine we start the vertexing, what happens, these vertexing will start agitation. Whenever agitation is started, what happens, these silica beads and other type of beads, what happens, it will percolate into the cytoplasm of the protoplast. This protoplast will make a four big pose because of the vertexing, and then desired gene will be enters into the location. That is called as the vertexing mechanism. And third, the seventh one is called as biolistics or gene gun method or particle bombardment method or ballistic method or particle gun method. In examination, whatever the things they asked, if they ask biologistic, gene gun, particle bombardment, ballistic or particle gene gun method, so you need to write only gene gun method here. So if you take the instrument that look like this, and it contains the a, a gas chamber. So this gas chamber fill up with some oxygen, nitrogen, whatever things are there. So those kind of cylinders will be inserted. So whenever that cylinder is inserted, this instrument also contains a chamber where gene of interest will be coated on the silver or a gold nanoparticle. Nanoparticle means you can observe that the size of the nanoparticle will be 10 to the power of minus 6 or 9. That ranges from 10 to the power of minus 6 to minus 9 type of size of gold nanoparticles or tungsten nanoparticles will be coated with desired DNA. Desired DNA will be inserted in this chamber. And then whenever gas chamber is released out a gas with high pressure, what happens? You can observe that. So that will project the gold nanoparticle or tungsten nanoparticle into the nucleus or cytoplasm of the plant cell. Even we can use protoplast, or we can use the plant with cell wall. So because of the pressure created, and then the projectile that is created for the tungsten or gold nanoparticle that is coated with DNA, that will directly enters to nucleus, and then integration happens successfully. So this is the percentage of the integration will be only 10 to 40% of the things will get hybrid cells here. Next method is called as electroporation. What is electroporation? What happens, in a soup of calcium chloride or in a soup of some solution EDTA or other type of chemicals are used. It act like a anionic buffers. So the cells, the protoplast cells with the, any desired DNA, whenever they enters into the a chamber that contains electric voltage. So the voltage should not be given in a prolonged period. You can observe that we can insert a voltage of 50 to 4000 volts per 30 to 40 milliseconds, not more than that. If you do that kind of electric field, that creates a transient pose on the cell membrane. Whenever cell membrane protoplast is inserted with the desired DNA in a soup, calcium chloride, polyethylene glycol solution, what happens? A small electric field is generated for some microseconds. What happens? It will cell membrane of the protoplast will create a transient pores, and through pores, this desired gene will enters into the cytoplasm, and then definitely that will integrates into the nuclear envelope. So this is where we can insert the desired gene into the protoplast cell of the plant here. This is one of the prominent method that is used here. This is the mechanism. You can see this is the steps here. Plant cell, remove the cell wall and insert in a solution that contains polyethylene glycol, and then we'll insert electrodes here. and whenever in small voltage is generated for milliseconds, a pore transient pores are formed, and then desired gene is integrates into the respective plant cell here. This is called as electroporation. And one more method, ninth method of physical method, was silicon carbide fiber mediated gene transfer. What is the method? So what happens here? Desired gene with target cell with silicon carbide is mixed in a solution that contains polyethylene glycol. What happens because of these silicon carbide fibers? This fiber will make a hole on a cell membrane. The protoplast, target cell protoplast will be there. On the protoplast you can observe that small pores are created because of this whiskers, silicon carbide whiskers. So what happens, the desired gene or DNA will be enters into the that pores created by these whiskers or silicon carbides. This is also a prominent method. So one more method is called as laser beam mediated gene transfer, where some voltage like 532 nanometer of the laser beam inserted for some 6 nanoseconds on the a protoplast cell. A protoplast of desired host plant cell will be exposed to laser beams of 532 nanometers for 6 nanoseconds. What happens, it will create a transient pulse, and then through that the gene of interest will be whatever with the vector gene of interest will be inserted, integrates into the host cell further. Next method is called as ultrasound mediated gene transfer. Same thing here. Instead of radiation, we'll use a high frequency of ultrasound for milliseconds. Again, it creates a rupturation on the cell membrane. Tiny ruptures are formed through which exogenous DNA or mRNA or other type of DNA will be inserted into the nucleus, and then they become hybrid cells here further. Next method is called as the selectable markers and antibiotic free transformation. Here we can use precise genome editing technologies called as CRISPR-Cas9 and talent technology, where precisely we'll edit the gene of interest in plant cell itself. When you design these pre-cRNA, whenever that will go to target site, it will modify it, it will enhance the expression, or it will knock out or knock in the genes of interest that is necessary for the formation of hybrid cells here. We can use both CRISPR-Cas9 and talent technology to edit the gene of interest in the host cell. One more transformation method is called as protoplast transformation. It's a common method where in a solution of PEG will make a protoplast, and then desired gene will be inserted in a polyethylene glycol, and then we'll use some chemicals so that will create a pores on that. We'll use liposome or other type of chemicals that contains the desired gene, and then further what happens, so transformation occurs, and then later on what happens, we'll produce a transgenic plants by protoplast transformation. And one more method in physical method is called a sonoporation. Sonoporation means we'll create a sound wave of some mega hertz for some seconds. What happens, it will create a micro bubble of vibration. This micro bubble of vibration, what happens, it will create pore on the membrane, and then because of that in DNA, whatever desired gene is there, modified genes with the vector will be enters cytoplasm, and then that will integrates into the nucleus also, and it forms the transformed cells further. This, and one more method, last method in physical method, is called as vacuum pump. What happens here, a desired plant's part will be inserted directly into the a vacuum chamber, and then a desired gene with the vector will be inserted on the surface of that. And whenever a vacuum is created, what happens, because of the vacuum pressure differences, what happens, this cell will expand, and then it forms a tiny pores on the surface of that. Whenever this the solution is dipped with the desired DNA, what happens, because of the vacuum pressure difference, so pore is formed, and then that will transfuse the gene of interest into the cytoplasm or nucleus of the desired plant cell. This is the 15th method of physical method. That completes the physical method. We'll go with a chemical method. You already know that, we'll use polyethylene glycol for transformation method, where the protoplast will be fused with the DNA and polyethylene glycol. Here, 10 to 15%, or it may ranges from the 10 to 15% concentration of polyethylene glycol, and wait for some time. It will be laborious and then time consuming. After some time, some plant cells, protoplast, will take up the desired gene and then become a transferred protoplast. That is the achievement of polyethylene glycol, but it consumes time. And one more chemical is used in chemical method is called as diethyl amino ethyl. So this is called as DEAE, dextron mediated gene transfer. What happens? A desired gene with this DEA will forms a complex, and it forms a liposome like a complex, and then through endosis cytosis process, it will take up to the take up to the cytoplasm. In cytoplasm, what happens? This will go and integrates into the plant genome, and then that will successfully converts that cell into transform cell because of the action of diethyl amino ethyl, DEAE. That is one of the prominent chemical used in the plant transfection method. So one more method in chemical method is called as calcium phosphate mediated. This calcium phosphate is a cationic type of chemical. Whenever they are mixed with the desired gene DNA, what happens? It will create a it will promote a pseudopodia type of structure in the protoplast, and then it will form endosome, and then the desired DNA will enter to nucleus and then helps in transfection. So calcium phosphate is also one of the prominent chemical that is used for the plant transformation method. And one more successful method that is used for the plant transformation is called liposome mediated. What happens, we'll prepare a cationic liposome where desired DNA is inserted. You already know that lipid molecules are there in the cytoplasm, in the cell membrane, that mainly make a way, easy getaway for the liposome that enters into the cytoplasm, and then this desired DNA will enters to nucleus, and then the plant cell become a transformed cell further. And one more chemical is used called as artificial lipids. Natural lipids also used, but in because of the scarcity, we'll also use some other effective type of lipids, artificial lipids called as lipofectomine, and then DOTAP, DODAC type of lipid. Artificial lipids are formed same way like liposome mediated transfer. These lipids also surrounds the desired DNA, and then by the action of endocytosis, that will reach up the nucleus, and then it becomes the transfer affected cells. And one more chemical called as protein polymer is also used. These polymers, cationic polymers, will surround DNA and helps in the formation of endosome, and successfully that will integrates into the nucleus of the host plant. And last one chemical used is called as dendrimer. This dendrimer mainly helps in attaching on the desired DNA. Whenever that will go and attach to the cell membrane, what happens? It forms, it will engulf by the process of endocytosis, and then same process by which the nucleic acid will be integrated into the nucleus and forms the uh, transformed plant cell. Next method called a second method is called the indirect method. Indirect method means we'll use a vector in between that successfully helps in the transformation. So here we'll use a vector called as a plasmid or Agrobacterium tumefaciens. Whenever you take Agrobacterium tumefaciens that contains TA plasmid, we'll modify a TA plasmid in such a way that it fit up the desired gene. Whenever desired genes are inserted into TA plasmid, it becomes a hybrid plasmid, and then whenever that infection happens, whenever any flower dips onto the solution that contains this agrobacterium that contains the desired DNA, what happens further? This flower will be transformed and then produce different types of hybrids further whenever you germinate it. This is a way in Agrobacterium tumefaciens, especially in case of the Arabidopsis thaliana, we can achieve this kind of the plant transfection. So whenever the flower inflorescence is dipped into a solution that contains Agrobacterium tumefaciens, that also contains desired DNA, definitely that will convert these seeds into genetically modified seeds here. Second method called as the Agrobacterium rhizogene, or we can also call as Rhizobium rhizogene, the current name. So this Rhizobium rhizogene, it contains a special plasmid called as RA plasmid. Agrobacterium tumefaciens contains TI plasmid. This Rhizobium rhizogene contains RI plasmid. What is RI means? RI means root inducing plasmid. TI means tumor inducing plasmid. What this Rhizobium rhizogene, what happens, it will induce more number of root hairs whenever they infected. When you compare that with Ta plasmid, the plasmid integrates into any plant. It causes cancer, tumor, and cause crown gall disease. Remember, so this Ra plasmid, you can observe that it contains this right border and left border. In between right border and left border, it contains the origin of replication. It contains virulence region that cause virulence effect, and then it contains also opine catabolism genes, and then even that contains some number of the hormones, so axin or gibberellin. So not axin, and then cytokinin genes also present, that helps in the formation of root, that is called as root inducing plasmid. It contains the hormones that will secrete vigorously and forms more number of root hair formation. So this way, will integrates the desired gene into the place wherever then this opine is not necessary, and other type of part is also not necessary. In that place, we can insert the gene of interest, and with a strong promoter will integrates into the plant genome with any methods. What happens, that will successfully integrates into plant genome, and then that converts into a transfected cells. And if you take the structural organization of the RA plasmid, it contains T region, tDNA. tDNA mainly helps in the integration process, and then virulence gene that mainly helps in the virulence effect of the Rhizobium rhizogene. And then opine synthase genes are there. This opine synthase will produce some carbon related compounds that drives the energy, and then replication, origin of replication will be there, through which replication starts. And then tra A and tra B genes are that mainly helps in the formation of sex pilars in conjugation. Remember, conjugation, this tra A and B genes are helps in conjugation and conversion of F plus in F minus into F plus. Remember, and rep ABC system is there. This that is a system where it helps in the replication of the entire gene. And then one more gene is there called gall operon. This gall operon mainly helps in the formation of gall or root induction in RI plasmid. Whenever desired genes are inserted in the place of virgin, definitely with the strong promoter, definitely that will integrate and then produce a transgenic plant. One more method is called Ti plasmid mediated gene transfer or Agrobacterium tumefaciens mediated gene transfer. Both are same because we need to insert the gene of interest into Ti plasmid only. So Agrobacterium tumefaciens is a natural engineer that mainly helps in transfer of the desired gene into the host, especially plant host. So this is restricted to plant host, not not applicable to animal host here. If you take the structural organization of Ta plasmid, it is also contains left border and right border. In between that, we'll have the coding gene for hormones, axins, and cytokinin hormones are secreted. Because of the secretion, what happens? It will produce a crown gall disease. Cancer cells are produced. Crown gall disease are formed in infected cells here, and even that contains opine genes, and we even that contains the virulence region. We can excise this region and insert our gene of interest and integrates into the plant genome, and successfully it will become a transfected cells or a transformed cell. So these are the regions here: oncogenes, axin, cytokine, whatever we discuss, that is present here. And how these Ti plasmid successfully enters into cytoplasm, even the nucleus are present, you can observe that here Vir genes are there. These Vir genes are take part in the proper infection and escape from the cytoplasmic nucleases activity. Whenever nucleus are present, definitely these genes will be degraded effectively. What happens naturally whenever the plant wound happens? Plant will secrete a chemical called as acetosyringon. So acetosyringon or syringon is produced. This acetosyringon mainly helps in the chemotaxis of these Agrobacterium tumefaciens. Whenever wound happens, this Agrobacterium that is freely present in the soil or in the air will attach to the wound region, and this acetosyringon will activate the Vir-A gene. Whenever Vir-A gene is activated, it will phosphorylate, and then what happens? It will activate in turn Vir-G protein. So genes are inactivated and then produces Vir-G protein. And this Vir-G protein, because of some other chemicals like glucose and other type of proteins, will activate a complex of Vir protein that is contains D1, D2, E2, B genes are activated. That is also related to Vir gene complex. Whenever this Vir complex is activated, that will binds to the tDNA, transfer DNA, and instead of transfer DNA, we can insert our gene of interest. You can see here tDNA region is there, right? In tDNA region, we can replace that. This axin gibberellin is not necessary, that causes disease. Instead of that, we'll cut here from the left border, right border. Our gene of interest will be inserted in the region of T regions. Sorry, I made a mistake here. Instead of wireless regions, this is necessary for the integration, successful integration. We'll insert the gene of interest into the T regions here. Whenever we'll insert our gene regions, these Vir D complex will binds to tDNA, and they forms a tDNA and Vir D complex is formed. So especially D2 complex will binds, and then it will entirely covers, and then it helps in the transformation. Whenever these complex are formed, it successfully escape from the plasmodesmata connection and then enters into plant cytoplasm. Here Vir-B acts in the transport of these complex into the cytoplasm, and further one more Vir protein will binds, and then it will surround this complex here. The tDNA Vir-D complex will be surrounded by Vir-E gene, Vir-E protein, sorry. So Vir-E binds, D2 binds, and then other protein complex binds, and that mainly helps in the transportation of these, whatever tDNA into the nucleus. Whenever tDNA enters here, it will go with a illegitimate recombination with the plant genome and helps in the integration of genome in the nucleus. In examination, they can ask that how, even though plant nucleus is present, this TA plasmid successfully enters and then integrates into plant genome. Definitely some question will be there in that these Vir complex will helps in successful transfer of the desired gene into the nucleus without the escape, which can easily escape from the nuclease activity of plant defense mechanism. Remember, so here Vir gene take part in successful escaping of these desired gene from the nuclease activity, other defense mechanisms here. So why Ti plasmid will serves as natural vector but not serve as a cloning vector? Cloning vector has some desired trait should be there, but in case of Ti plasmid, because of large size, they cannot accommodate. Only they can accommodate from, you can see here, so from left border to right border, definitely that is around 20 to 4 base pairs, and then the internal some limited base pair is there, and all other things are necessary for the Ta plasmid to integrate successfully. So because of that, so it has larger size, and because of that condition, it cannot integrates the foreign DNA. And one more thing is, it lacks the specific restriction sites. So you must know that to integrate into the particular plant genome, they must have some restriction site, otherwise the restriction enzymes will not work and splice it. BAM3, ECO RY, R1 type of restriction enzymes are there. Here they lack the restriction enzyme site. And even this plasmid, TA plasmid will have a tissue specificity, only stems will be infected, not the leaves. And then even the TI plasmid is very complex here. And even that contains phytohormone genes that should be removed. Otherwise, definitely we cannot accommodate more genes here. Even opine genes are not necessary, that should be removed. And then even marker genes like ampicillin resistant genes, tetracycline resistant genes are not there, not here naturally. Because of that, it will not act like a cloning vector here. Even it is not able to replicate in some organisms, especially in E. coli. Remember, in E. coli, even though that is bacteria, this is also bacteria. So this it will not replicate. TI plasmid will not replicate in E. coli. So because of that, wherever transfection happens with respect to E. coli, TI plasmid will not replicate it. Remember. Next, one more type of virus mediated gene transfer is cauliflower mosaic virus. This is double stranded virus. It contains around 8,200 not 8,024 base pairs are present, that around 80 kb, sorry, 8 kb, capacity is there. If you take the double stranded DNA, circular DNA of the cauliflower mosaic virus, it contains it segregates into 7 different regions here. In that, first region is there, second, third, fourth, fifth, sixth, fifth, sixth, seventh region is there. 1 2 3 4 5 6 7 8, sorry, eighth regions are there. In that, only it will accommodate the genes, the first 3 4 8 5 and then 6 is essential, but 7 and fifth one is non-essential region. So sorry, seventh and second is not essential for the transformation. So in this region, small region, we can insert our gene of interest, but the only drawback is the whatever the gene desired gene should be limited. We cannot accommodate bigger DNA in case of cauliflower mosaic virus. So one more drawback is that this cauliflower mosaic virus is restricted to Solanaceae family. That means cauliflower, tobacco, and other type of members, or even some cauliflower families only they will infect and causes diseases. Because of that, remember, this virus is this virus will accommodate less gene of interest, and then only some Brassicaceae family only it will infect, not Solanaceae, sorry, that's my mistake. It will only infect some species of Brassica members here. And one more virus is rice tongue virus. If you take this rice tongue virus, it is also double stranded virus. It is also almost similar, 8,000 base pair is present. It has nearly four open reading frames are there. If you take the open reading frame, so it contains different coding parts, origin of replication, and other part here. In that, you can observe that some regions of intergenic regions, and then this open reading frame 4 is not at all necessary. So we can accommodate this space for our gene of interest. That's it. So in this case, this open reading 4 and then some intergenic regions are we can remove it and then insert our gene of interest. Other than that, other reading frames are essential for us. Next, we'll move on to one more virus called Gemini virus. Gemini virus is called, one or two single-stranded circular DNA is present. Single-stranded circular DNA, ssDNA. So here it is a small circular DNA is present. It can infect different types of monocots and dicots, but the accommodation of genes will be limited here in case of Gemini virus here. One more virus called tobacco mosaic virus. Here also it is single stranded sense RNA molecule. If you observe that it contains different open reading frames, nearly 1, 2, 3, 4, 4 open reading frames are there. In that, it contains around 6,400 nucleotide long single stranded RNA molecules are present. it contains, it will express four major protein that helps in replication, replicase protein. Second one is called as movement protein that helps in the movement of this gene of interest, and third one is code protein, and fourth one is RNA silencing protein. Here we can the replicase protein should be necessary, movement protein should be necessary. We can remove the code protein, and then in that place we can easily accommodate our gene of interest, and then even RNA silencing protein is necessary. So these three proteins are necessary. These genes should be retained, and then we can remove the code protein, and then we can integrate our gene of interest. But limited genome will be inserted in tobacco mosaic virus. Next virus is called as bromo mosaic virus. It is also same as that of the tobacco mosaic virus. It contains a tripartial, that means three circular RNA genome is present. You can observe that it contains RNA, RNA1, RNA2, RNA3. But the drawback is that it can accommodate less genome. But one more advantage here, it will affect or infect wide range of the plant species here. You can see RNA1, RNA2, RNA3 is there, and RNA4 is there. Some will code for a helicase, and some will code for cell-cell movements, replication, and this region is not necessary for us. We can remove, and then we'll place our gene of interest, and we can easily accommodate and then convert that into a transfected cell.